Welcome back to BioB 160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and in this episode we'll explore the fascinating story of DNA, the double helix and ladder of life. Let's get started. DNA was discovered in the early 1900s by Friedrich Miescher. He isolated a chemical from the white blood cells in pus-filled bandages that he called nuclein. This was later renamed DNA, deoxyribonucleic acid. Miescher found that DNA was a major component of the cell's nucleus, but it had relatively simple chemical makeup, just 4 different chemicals called bases. It seemed too simple to be the chemical that could encode genetic information. On the other hand, proteins are made of 20 amino acids, and that seemed like a more likely candidate as the cell's genetic information. So what is the genetic information of the cell? To review what we've discussed so far, we know that Mendel observed patterns of inheritance in pea plants, and he used those observations to develop his laws of heredity. Thomas Hunt Morgan studied fruit flies and was able to demonstrate that genetic information was carried on chromosomes. In the early 1900s, many scientists were convinced that the units of heredity were carried on chromosomes, but they had no clue about the molecular mechanism of inheritance. They knew that chromosomes were made of 2 types of chemicals, protein and DNA. But which one was the genetic material of the cell? Was it DNA or was it protein? The story of the search for genetic material of the cell spans several decades, from the 1920s to the 1950s. To understand this journey, we'll look at 3 key experiments. These are Griffith's discovery of transformation in bacteria in 1928, the work of Oswald Avery and his co-workers in the chemical basis for transformation, and the work of Alfred Hershey and Martha Chase on the DNA of the bacteriophage T2. Frederick Griffith didn't set out to discover the role of DNA in the cell. He was studying the bacteria that caused pneumonia, Streptococcus pneumoniae, and was trying to develop a strain of bacteria that could be used as a vaccine. To develop his vaccine strain, he isolated the virulent or disease-causing Streptococcus, and he cultured the bacteria until they lost their virulence. His virulent strain produced a capsule around the bacterial cell, and the cells looked smooth when they grew on agar. He called the virulent strain the S strain for smooth strain. The avirulent or non-disease-causing strain did not have a capsule and would look rough when it grew on agar plates. He called this strain the R strain for rough. In his experiment, he began by injecting the virulent S strain into mice. As expected, the mice died from infection. If he injected the avirulent R strain into mice, the mice remained healthy and survived. When he injected heat-killed S strain bacteria into mice, the mice also remained healthy and survived. The unexpected result came when he combined the living R strain bacteria and the heat-killed S strain bacteria, and the combination of these 2 caused an infection that killed the mice. In examining the mice that died, he found living S strain bacteria. How had the living R strain acquired the capsule from the dead S strain? Griffith did not know the answer, but he called this transformation. Something about the dead S strain had transformed the R strain so that they were now S strain bacteria. So what substance from the S strain could transform R strain bacteria into S strain? Oswald Avery set out to answer this question. He worked with his colleagues Macklin McCarty and Colin McLeod and made cellular extracts from the S strain to see what chemical component might be responsible for the transformation. He repeated Griffith's experimental mixture, but instead of using dead S strain cells, he used either the polysaccharides, the protein, or the DNA from the S strain bacteria. Their experiment showed that mixing polysaccharides or proteins with the live R strain did not cause transformation. However, when they mixed the DNA from the S strain with the live R strain, they observed that the mice developed infection and died. They were also able to discover live S strain bacteria from the dead mouse, just as Griffith had found in his experiment. Their conclusion was that DNA is the material that causes transformation of the R strain into the S strain. Avery's work was a big step towards proving that DNA is the cell's genetic information, but many scientists were not convinced. Another pair of investigators tried a different approach. Alfred Hershey and Martha Chase were interested in viruses, particularly the bacteriophages. Bacteriophages are viruses that infect bacteria, and they're very simple infectious agents that are made of just DNA and protein. Hershey and Chase set out to ask a very simple question. What part of the phage actually enters the host cell and causes the infection? Is it the DNA or the protein? To answer this question, they used radioactivity to label the proteins or the DNA. The bacteriophage proteins were tagged with the radioactive element S35. Since proteins have sulfur atoms and DNA doesn't, all of the radioactivity ends up in the proteins. To label the DNA, they used P32. Again, phosphorus is found only in DNA and not in proteins, so all the radioactivity ends up in the DNA. They set up 2 batches of host E. coli cells and infected one batch with S35 labeled phage and the other batch with P32 labeled phage. In each case, the phage attached to the host cells, and then a blender was used to separate the phage from the host. Next, they centrifuged the culture solution and tested to see where the radioactivity was located. Was it with the pellet of the host cells or the supernatant? In the batch with labeled protein, they found very little radioactivity in the cell pellet and most of the radioactivity in the supernatant liquid. This indicated that none of the protein entered the host cells. In the second batch, they found nearly all of the radioactivity in the cell pellet. This indicated that the DNA from the phage entered the host cell and was responsible for the replication of new viruses. So the next big question was, what is the structure of DNA? The basic makeup of DNA was known, but since DNA molecules are so large, no one knew exactly how the parts were put together. Erwin Chargaff studied DNA by extracting chemical bases and analyzing the exact proportion of the bases in a variety of very diverse organisms. These included yeast, wheat, salmon, and humans. He found that the base composition varies between species, but it was constant for a given species. He also found that the ratio of adenine to thymine was always 1, and the ratio of guanine to cytosine was 1. He called these ratios Chargaff's rules, and this meant that the percent adenine was equal to the percent thymine, and the percent guanine was equal to the percent cytosine. This still didn't answer the question of the structure of DNA, but it was a very helpful clue. In the mid-1950s, several research groups were working very hard trying to unravel the structure of DNA. 4 of these investigators are shown here: Rosalind Franklin, Maurice Wilkins, Jim Watson, and Francis Crick. Franklin and Wilkins worked at King's College in London and were developing methods to crystallize DNA and use X-ray diffraction to examine its structure. Watson and Crick were young researchers at Cambridge University and were attempting to solve the structure of DNA by building models based on information gleaned from various sources. This photo, taken by Rosalind Franklin, was an important clue to understanding DNA structure. Her photo showed that DNA is a helix, and the spacing of the atoms in the helix could be measured by a careful examination of the photo. With this piece of information and knowledge of Chargaff's rules, Watson and Crick built a model for the structure of DNA. About the same time, the American chemist Linus Pauling also attempted to solve the structure of DNA. Linus Pauling was a very famous protein chemist who was awarded the Nobel Prize for his work on the alpha helix structure of proteins. Unfortunately, Pauling had very poor X-ray crystallography data, and his model was not correct. However, his incorrect structure of DNA was helpful to Watson and Crick, and since they had Rosalind Franklin's photo showing the helix structure of DNA, they were able to build an accurate model of DNA. Watson and Crick published their paper in the journal Nature in April 1953, and their paper was immediately followed by a paper by Franklin and Wilkins. Interestingly, since Watson and Crick built the first model of DNA, they are always credited as the discoverers of the structure of DNA. I'll leave it for you to decide who you think should be credited for determining the structure of DNA. And if you're interested in learning more about this fascinating chapter of science history, watch the PBS Nova episode, The Secret of Photo 51, which gives a biography of Rosalind Franklin and describes her role in the DNA story. This is for a little extra credit work. There'll be an extra credit question on the next exam on this topic. Go to the Nobel Prize website and find out who's shown in this photo and why each of these individuals were awarded a Nobel Prize in 1962. The double helix model of DNA that was proposed by Watson and Crick and Franklin and Wilkins is shown here. DNA is made of 2 strands of nucleotides that are wound together, making it look like a spiral staircase or spiral ladder. The backbone of the helix are sugars and phosphates, while the rungs of the ladder are the bases, adenine, guanine, cytosine, and thymine. The base pairing in the ladder is consistent with Chargaff's rules and is due to the structure of the bases. Adenine always pairs with thymine. If you look at this diagram, you can see that adenine and thymine have 2 places for hydrogen bonding. Guanine always pairs with cytosine, and this is because they have 3 places for hydrogen bonding. The base pairing also relates to the width of the ladder. DNA has a constant width, and this requires that a purine, either adenine or guanine, pairs with a pyrimidine, either thymine or cytosine. If two purines pair together, the structure is too wide. If two pyrimidines pair together, the structure is too narrow. But a purine paired with a pyrimidine is exactly the right size. So how are the nucleotides joined together in DNA? As shown here, a single nucleotide consists of a base, one of the four bases A, T, G, or C, joined to the sugar deoxyribose and a phosphate group. Nucleotides are joined by linking the phosphate of one nucleotide to the sugar of the next nucleotide. Notice that the phosphate group is always on the number 5 carbon, called the 5' carbon, of deoxyribose. Also notice that the phosphate always links the number 3 carbon, called the 3' carbon, of deoxyribose. This forms a 3' to 5' phosphodiester linkage. Also note that each strand of nucleotides has directionality. At the top of the diagram you can see a nucleotide with a free 5' phosphate group. This is called the 5' end of the strand. At the bottom of the diagram you can see a free 3' hydroxyl group on the deoxyribose. This is called the 3' end of the strand. The 3' and 5' ends of a complete DNA molecule can be seen here. What do you notice about the ends of the two complementary strands? If you look carefully, you'll notice that one strand with a 5' end pointed up is paired with a strand with its 3' end pointed up. The opposite is true of the bottom ends. Each strand has a 3' and 5' end, but the two strands are running opposite or anti-parallel of each other. We'll be talking more about 3' and 5' in our segment on DNA replication, so it's important to study this diagram to understand what these terms are referring to. This diagram shows a summary of the important features of DNA. It is a double helix consisting of two strands of nucleotides wound together. The backbone of the double helix are the alternating sugars and phosphates, and the rungs of the latter are the purine-pyrimidine base pairs. And these base pairs are held together by hydrogen bonds. And the polarity of the two strands is opposite or anti-parallel to one another. This concludes part 1 of the molecular basis of inheritance. In part 2 of this episode, we'll look at DNA replication.